All right, welcome back to our discussion of bioenergetics and metabolism, and we're going to go ahead and dive right into the glycolysis pathway. So, our learning objective here is to summarize reactions of glycolysis. So, first thing to put this into perspective is here is a chart kind of laying out all of the major pathways of biochemistry. So we have all of our various macrobiomolecules such as lipids and proteins and nucleic acids. And this is all tying together how things fit together. Notice running right through the middle of this is glycolysis, right? And that extends directly into the citric acid cycle. So these two progressions act as the central hub of metabolism, which is why we spend so much time talking about it. Because if you understand the central hub, it's easier to see where all of these other side channels come into. So looking at this from a superficial level and think about where things actually happen, here we have glycolysis, where we go from glucose to pyruvate. This is all happening in the cytoplasm. And in this step, we gain electrons that we can then transfer, but we also gain some ATP along the way. When we progress towards the citric acid cycle, which is taking place in the mitochondria, we go through the substep where we turn pyruvate into acetyl-CoA. Again, this is happening within the mitochondria. This process here, the citric acid cycle, is going to give us more ATP from a substrate level, and we'll talk about what that means here in a second, but also more electrons. Ultimately, those electrons get dumped into this oxidative phosphorylation pathway, or the electron transport chain, and that is what leads to the bulk of ATP. And this ATP we refer to as oxidative ATP, because this is essentially taking those electrons, gleaning energy from it, and using that to make ATP. So oxidative phosphorylation accounts for a good 90% of the ATP that is generated. And again, this is powered through a chain of redox reactions, okay? But there is a smaller amount of ATP that is formed in glycolysis and the citric acid cycle that occur through something that we call substrate level phosphorylation. And how this is different is this is ATP that is generated by enzymes working along the steps of this process, right? So it's not making a lot, and it's not quite the same as what's happening in the oxidative process, which is more like a ATP factory, so to speak, and we'll get into much more detail on that. This is more of a along the way, hey, you can do this, okay? And this can provide energy when you are under an oxygen-starved scenario. Now, for each molecule of glucose that's degraded to CO2 and water through this process, the cell can make up to 32 molecules of ATP. And this isn't an exact number, and we'll talk about why it's not a fixed number a little bit later. All right, so we're going to focus in now on the central part, this glycolysis pathway that sits right here at the hub. And I'm going to go through the reactions in detail, kind of pointing out some features along the way. Now, glycolysis is broken up into two parts. One is an energy investment phase where we actually burn ATP. okay? We'll then have a second part, which is called the energy production part, where we can glean back that ATP with some, and then we also gain the electrons along the way. And those electrons can have their own fate. All right, so this is all starting out with glucose. And something that I want you to keep in mind, and we'll talk about this more, is we're using glucose as an example of how things progress through this pathway. Other sugars other than glucose can proceed through this pathway through slightly different branches. All right, so first we have this glucose molecule. It has 6 carbons, 1, 2, 3, 4, 5, 6. The first thing we are going to do is phosphorylate it. Key point here is phosphorylating it effectively reduces the presence of glucose from an equilibrium standpoint. Okay, so if we keep turning glucose into glucose 6-phosphate, then we can keep shifting the equilibrium towards making this reaction run. Okay, and we can continue to pull in glucose from outside the cell. So this is the first step here. The next reaction goes from glucose 6-phosphate to fructose 6-phosphate. The key difference here is we are just really reshuffling the bonds. So it's the same formula, so to speak. We're just reshuffling the bonds. And notice what's happening here is we go from something that is asymmetrical to something that is very symmetrical. Okay? Keeping in mind that we're ultimately going to be splitting this into two, you can see why having it symmetrical would be beneficial. Now, if you look here, what's the key thing that makes this less symmetrical? Well, the side is missing a phosphate. So, naturally, we add a phosphate. So again, we're building symmetry, and then we can take this molecule and cleanly split it in half, all right? Now, this doesn't split into two exactly identical molecules. It has some minor structural differences. Fortunately, that is something that is easily rearranged so that we can take this compound here, dihydroxyacetone phosphate, and convert it into glyceraldehyde 3-phosphate, which can then progress on. Coming back over here to the energy production side, we've taken a 6-carbon compound, we've now split it into 2 3-carbon compounds. So recognize that everything happening on this side is actually happening twice for every single molecule of glucose. So one thing that we can do is we can add on a phosphate group, and this is adding on an inorganic phosphate group. This requires a major electron activity in order for you to pop this phosphate on. Phosphate groups really don't want to be attached to things. So how do we achieve that? Well, we use a compound called NAD to strip electrons from the molecule, which forces the phosphate ion to attach on or to fill that role. Since it is very electron rich, if we take electrons away, we now have something that can act as a nucleophile, right? So what is NAD? NAD is a very complex molecule, and it is built off of ATP, right? So here we have the nitrogenous base, here we have ribose, we have a couple of phosphate groups, then we have an additional ribose, and then we have this thing up here at the top that is a nicotinamide group, okay? This is actually the business end of NAD, okay? This is what's doing all the work. The rest of this is actually just a handle, right? So like we the cells have evolved to focus on ATP because we have very good ATP binding domains that get replicated, and evolutionary selection kind of draws on that. The idea of using ATP simply as a handle for this, what is in reality a very small group, um, makes this work. So this is something that an enzyme can bind. This is the functional group that actually does work. So how does this actually work? If we have a hydrogen ion attached to another molecule, we can essentially utilize the electrons from there to attach onto here. We get some electron reshuffling, and we attach that hydrogen on, right? I'm not worried about you guys understanding this aspect of it, but recognize that when NAD takes something from a molecule, it is taking a hydrogen with both electrons, right? So one electron which belonged to the hydrogen and another electron which it was sharing in its covalent bond. It takes both, okay? And that has consequences on the molecule that it takes it from. So coming back here, we strip a couple of electrons. This now takes G3P and turns it into this 1,3-bisphosphoglycerate, which now has two phosphate groups. We then can pull off one of those phosphate groups and regenerate ATP. Since this is happening twice, we've essentially recovered both of these ATP molecules. Next step down here, we're just going to do some rearrangement to make slightly different molecules. Again, just reshuffling, until we end up with this molecule here. This molecule is actually one of the most energetic molecules that we have in our body. This is actually has more potential energy than ATP itself. And that has to do with the double bond that's existing here adjacent to this carbon with the phosphate group. So, being such a high energy compound, we actually have the capacity to add that phosphate directly onto ADP, gaining to ATP along the way, and this leaves us with pyruvate, which can then go on to the citric acid cycle. As a side note, we can take pyruvate and do some other manipulations to produce either lactate or ethanol, depending on what kind of organism you are. So yeast produces ethanol, and we and other organisms can produce lactate. The only reason why we actually do this is if we are in anaerobic conditions where there isn't oxygen, and what we need is to regenerate the NAD so that we can at least keep this part of the pathway running. Without NAD, it can't run. So here we're essentially just taking those electrons and dumping them back onto the molecule itself so that we can recapture that NAD. Now, this series of reactions have some spots where you get major changes in energy, okay? So one is this step from glucose to glucose 6-phosphate. Another one is happening here. And another one is that final step. These are non-reversible reactions, okay? To where in the middle, we actually have a whole series of reactions that are reversible. And I'll pick this up in part 2.